Why don't we set it up? Give me 1 second. Where is the volume? Oh, okay. The meeting has been live streamed right now. It's set up.
Sit,
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 Streamed right now. It's set up.
sit.
Streamed right now. Streamed right now.
Okay, so it is working fine now. So it is working. So it is working fine.
Something is wrong. I see your audio many multiple times.
Yeah, I think there is probably because I was watching it, right?
Oh, you are
The
watching
live.
it. Okay, okay,
Yeah,
okay.
I was just
Perfect.
checking if it is working or not.
Okay, makes sense. Makes sense. Makes perfect sense. Okay, so this is good. And I think we are good to go. So we'll wait for 9 minutes. And I sent people the link of WhatsApp. If nobody joins, we'll have it recorded and live streamed. But people should join. Yeah. Give me 1 second. I'm gonna email them as send them via WhatsApp as well. Yeah, I'll join in 1 minute. Okay, I'm taking. Yeah.
Yeah.
Yeah. Hi, everyone. Thank you for joining. We'll be starting promptly in like 4 minutes. Dr. Gupta, I sent you, since I give you the host rights, I sent you. Perfect. Hi, everyone. We'll be starting in promptly 1 minute now. Can everybody see my screen?
Yes.
Okay. We have few 5-10 seconds more remaining, I guess, and then we should start promptly at 9. By the way, for people who are joining, this meeting is also live streamed now in Dr. Gupta's YouTube channel. Hello.
Yes.
Okay. Okay, I'm going to start now. So, hi everyone. Welcome to this interesting session for DNA storage, opportunities and challenges. We have Professor Manish Gupta, who's part of the Dhirubhai Ambani group and a part of also the Gupta Lab. And this is our 17th session. So, before I start, I'm the host. My name is Taposh Roy. I'm a director of innovation at Kaiser Permanente. My focus is on digital twin, machine learning, and anything innovation. I'm the author of a couple of books, Intelligent Governance for AI and both classical and quantum areas, and then medical image processing with deep learning. Um, Today we have a volunteer. She will be online pretty soon. She is a middle schooler in San Ramon, California, and she's going to help us with the survey, QA, and everything else. Um, the speaker today is Dr. Manish Gupta. Manish Gupta has earned his PhD in mathematics from Indian Institute of Technology in Kanpur. He has held several academic positions. He's been a professor at Dhirubhai Ambani Institute of Information and Communication at Gandhinagar since 2006. He is one of the leaders in the DNA storage, and we are really, really happy that he has lent us his time to come and talk to us about this amazing technology and how this can help in the future. So I don't want to be a barrier between you and him. I'm going to stop sharing and I'm going to request Manish sir to take us through the journey.
Thank you, Dr. Tapa. So let me share my screen, and if it is visible, can you just let me know if the screen is visible?
Yes, it's visible.
Okay, if there are some people waiting, I guess. So, okay, so I think the screen. So first of all, good morning in US, and good evening everywhere else. I guess I mean in India, good evening, and I don't know, people might have joined from different places. So good morning, good evening, good night. Anyway, so this is a really interesting topic which I'm going to talk about today. So I'll take about 30 to 40 to 45 minutes about a new technology that is emerging, known as DNA storage. And I should say that this area of DNA computing, actually in the year of 1994, something really interesting happened. I think you might be knowing that since I'm talking to the quantum computing group. The Peter Shaw discovered the integer polymer time integer factorization and discrete log problem. That was a really important year, and I think I still remember when this paper came out in 1994. I was doing PhD at IIT Kanpur at that point of time, and at the same time, there was interesting development that was done by Edelman. Edelman, I think some of you might be knowing that he is one of the uh, co-founder of RSA Crypto Systems. Okay, so he was reading the books on biology, and then one day he realized that these DNA this DNA and protein these kind of things can be used for computing actually, and he planned an experiment of solving traveling salesman problem, a small problem actually, instance of that, using bunch of DNA strings. And the key idea about DNA computing was DNA hybridization, because you know that DNA is ACGT, so A is complement to T and C is complement to G, right? So that was the backbone. And I also entered in this area because whenever you want to make computer, whether it is a quantum computer or it's a DNA computer, you require error correction, right? So that's how I entered into this area. And since then, this area has gone into different directions. The DNA computing. And so initially, I'm going to talk about some of the initial development, some historical development in this area, and then I'll focus on this DNA storage actually, what has happened in DNA storage. The area of DNA storage started something around 2012, I would say. Of course, there were some early ideas before that, but the first experimental work was done in 2012 and then 2013. So George Church at Harvard, actually, he did the first experiment in 2012. After that, Nick Goldman, they did improvement on that algorithm, and they were able to store petabyte of information into 1 gram of DNA. Nick Goldman was at Cambridge at that point of time, and after that, we were luckily third, to do work in this area, and we actually modified Goldman's algorithm. We implemented it, and we realized that it can only implement up to 500 MB. It can only encode 500 MB file. So we improved the algorithm, and our algorithm went a bit further, and we reached really exabyte order actually. So that was the improvement, and that's how we entered into the DNA storage. And then luckily I got one PhD student who started working in this area. and we worked on different directions in this area. Anyway, so the picture that you see here at in the middle is actually what when we have done experiment with DNA storage, and this was a demonstration to the Prime Minister of India and Prime Minister of Israel in 2018. Anyway, so let me start. So I'll start with this motivation, which is given by Edelman. So after this discovery of DNA computing, he mentioned this that biology and computer science, life and computation are related. And I am confident that at the interface, great discoveries await those who seek them. So, and in fact, this turns out to be really very important statement that no matter what portion of biology I see, I can clearly see a computational part. And if you look at computer science as well, you can get a lot of interesting ideas from biology as well. So it's really very, very, interesting thing. And all this happens because when we look at the world around us in the form of information processing, right? So this is something really very, very important understanding. And Seth Lloyd at MIT has said that our world, he gave that what kind of information processing principles are around us, whether you're looking at brain or atoms or universe as a whole. In fact, he argued that in his famous book that universe is nothing but a big quantum computer, right? So those are some interesting things when we look at when we mix computer science and biology. So now, now there are certain commercial aspects of these new emerging areas. So as you can see that CRISPR is one of them, and DNA storage is people expect that by 2028 it will be around 1926 million dollar. In that range, there will be market for this, and of course, there is a synthetic biology as a whole, quantum computing, drone, and all kind of emerging technologies. So it is one of the gem in this emerging technologies, and so I'll show you actually, what are the where this right now the technology stands and why we need this DNA storage. This is something really important, and you will see that every day we are generating data, right? We are clicking photographs, uploading on Facebook, social media, this and that, and how much data we are generating. I'll quote a statement by Eric Schmidt, who is the former executive chairman of Google, that around the year 2010 we were actually generating about 2-3 exabytes of data every day, right? So it's really exabyte is the order of 10 is per 18. I'll talk about these terminology pretty soon. So basically, let us talk about some brief history. So this biomolecular computing, is a new area when we mix biology and computer science, and there are 5 disciplines right now people are studying. Membrane computing, which is more theoretical. Peptide computing is when you want to make computer out of proteins. RNA computing, DNA computing, and bacterial computing. So in fact, a lot of experimental work has been done on DNA computing and bacterial computing. Okay, so these are the 3 topics which are more on experimental sides. Membrane and peptides are more theoretical, from theoretical computer science perspective, people are studying it. Okay, so what is DNA computing? So, when we are computing with DNA strands, so on the top row, you see there is a computational problem, and it is solved. A bunch of, binary strings are actually solving it using some gates and circuits. That's what you see, the computer that we use today. On the other hand, any computational problem, you can convert into the bunch of DNA strings, and those DNA strings actually interact with each other. And in fact, there is a new area which is known as, uh, DSD, dynamic strand displacement of DNA, right? So a bunch of DNA strings are actually interacting with each other. I call it as a dancing and actually solving a problem. So those kind of things are also people are doing. So anyway, so finally, output is also in the form of DNA string. So you can read the solution in the using certain DNA operations, which are normal molecular biology lab techniques. So this is something that is analogy between the two. Okay, now if you talk about some of the applications that people have come up with, of course, there is a DNA tile is an approximation, a theoretical model for studying the DNA computing, and DNA nanostructures is one nice area. So you can actually create things out of DNA molecules. In fact, one very popular area is DNA origami, which was started by Paul Rotman in 2006. He from Caltech. It's a really nice area. So for DNA origami means you're folding DNA into the desired shape. So you can make maybe a small box, or maybe a small artificial virus or vaccine. All kind of things are possible there. Then there is a DNA cryptography. I mean, that area has emerged a lot actually. DNA circuits is there. You can make a lot of circuits. DNA strand displacement, as I talked about, and DNA tiles are basically a theoretical model, but of course there are some implementations as well for those things. But today I'm going to talk about more about DNA data storage. Okay, so which is this. Now, storage is the fundamental need. Now, I'll tell you something, how much data we are actually generating. Storage is a fundamental need for both life and computation, and it's a very, very basic computing primitive. And you can think that life is nothing but a storage for plus information person, which is computing. And modern humans stores data. Now, what kind of devices that we are using for storing the data? Now, you can see that this is another example of natural storage. So nature has, these are known as honeypot ants, honey. Anyway, so now you see from cave paintings, stylistic papers, paper, punch card, hard disk, RAM, floppy. You might be familiar with some of the people who are of bit older generation. They might have seen floppies. I don't know whether the young people have seen it, but then CD-ROM, DVD, flash drive, Blu-ray disc, HD DVD. And nowadays, whenever you log into a Gmail account or so, actually you're using cloud storage. So this is basically sort of a historical development, how storage devices have evolved. And now one can ask, so I actually, in a couple of minutes I will convince you that DNA storage is the ultimate solution for our storage need. So first of all, we should know how much data we are generating. Now look at in 1956, this is the first hard disk drive, IBM 350 model, and it used to store, just under 5 MB data, and see how large, in a plane they used to keep it. So it's something really interesting. I'll show you another with capacity 1 GB in 1980. This was also very large. Okay, and I think similar kind of machines are available right now for DNA storage things. I mean, the company spin-off from MIT, Catalog is actually working on similar machines at the moment, for storage. Okay, uh, so now you see another comparison between. I mean, I think this is just similar to quantum computing and uh, you know and the classical computer, so IBM computer in 1958 and 2021. So similar kind of things. You see the. So what I'm saying that, so I want to convince that in the early days, all these machines, whether you talk about computer or you storage device, they are very bulky. And over the years, people come up with, machines which are a bit smaller in size, and then they are quite handy for use. So there are some heroes of storage technology. Keys Imic, I mean, he has more than 1,100 patents, all digital audio, video, CD, DVD, you file patent from him. And he has one company, Turing, which he's from Holland. And Samuelson who is one of the inventor in pen drive. He has also done a lot of great work. In fact, he is also, some of you might be knowing that he is also, the sort of founder of DNA, founder of semiconductor, so biological semiconductor technology. This is something, a very interesting thing. Now I'm going to talk about elephants, means data. So you see that I'm talking about big data. So how many elephants we are generating? So this is the statement given by Rick Smith. From the dawn of civilization until 2003, humankind generated 5 exabytes of data. 1 exabyte is 1 billion gigabytes, in the order of 10 is for 18. And now we produce 5 exabytes every 2 days, and the pace is accelerating. By the way, this is a statement he gave in 2010. And right now, so one can ask, and we try to estimate the size of internet. It's something around 700 to 800 exabytes of data, of the current size of the internet. So every day we are generating a couple of exabytes actually. And so obviously, if we store so much data, it costs a lot to the companies, right? Especially Facebook, Google, all these companies who are actually generating this data. And in future, one can see that the cloud of Internet of Things will produce data in the order of 10 to the 30 zettabyte. So this is a snapshot of 60 seconds. It's a bit old one, but it gives enough understanding that how much data we are generating actually. And so basically, you might have heard megabyte, gigabyte, terabyte, petabyte. So exabyte is what I'm talking about. Then zettabytes is for 21. Yottabyte is around 250 trillion of DVDs. Brontobyte and gigabyte. So now actually I will, if we actually suppose we want to store 1 yottabyte of data, so in the current technology, if we use the current technology, it is going to take a lot more space, and also it is costly. So let us see. If you want to store yottabyte, it will require, millions of data centers in Delaware and Rhode Island in US actually. This is how a comparison on the left hand side you can see. And again, although this table is a bit old, I have 2008, you can clearly see that the world GDP was $61 trillion, and at that point of time, the cost of storing 1 yottabyte was $100 trillion. So really, this is very expensive technology that we are using right now. Uh, you know, and so obviously there is a lot of other overhead, for example, electricity and so on and so forth, right, for storing huge amount of data. This is a really burning problem. So, and that's why industry is convinced. And in Silicon Valley, there is a SNIA group that was formed by a lot of companies, especially Microsoft, Twist Biosciences, Illumina, and Western Digital. So these were the founders, and now there are around 40 people who are member there. We are also member there. So we are contributing. So I'll talk about it in a minute. So now the question is, what is the solution? What is the ultimate storage device? So it turns out that 1 gram of DNA can store 455 exabytes of data. So ultimate storage density, and that means maybe a couple of grams of DNA can store in the entire internet that you see, right? And it is expected that 1 kilogram of DNA can store anything ever produced on this planet. All kind of data. So this is really ultimate storage device. And obviously we would like to see a pen drive like this. You just plug in into your computer, data is stored, right? So I'll talk about it, what kind of challenges are there in actually getting this kind of technology, right? So now something about the DNA. So now you can see that there is it's made of ACGT, adenine, cytosine, guanine, thymine, right? And the A and T and C and G are complement each other. You can clearly see that there are 2 hydrogen bonds between A and T, and there are 3 hydrogen bonds between C and G. And this hybridization is a very, very important thing. It's a basic. So basically, when we convert data into basically when we write data into the DNA, we use a machine known as DNA synthesizer, and there are three approaches available right now in the market. And the other hand, because of the Human Genome Project and a lot of advancement in biotechnology, you can see that we have Oxford, we have a nanopore sequencing, which is very, very small sequencing devices are available, means for reading the data, right? A lot more progress has happened there. And right now, there are a couple of companies which are working on, efficient DNA synthesis as well. Okay. So, okay. So now, if we want to store data into the DNA, one thing, there are two approaches right now. I will talk about it. In fact, in this talk, I will talk more about sequence-based storage. So basically, you are converting the byte strings 0, 1 into the DNA using some sort of mapping. Okay, and so that this mapping satisfies certain biological constraints, and you can create efficient algorithms for error correction and so basically data encoding and decoding. There is another approach. Of course, there are certain limitations with sequence-based storage because this is more popular for archival storage. On the other hand, there are structure-based storage in DNA, and this is just like close to our hard disk that we use. You can rewrite, in the structure-based DNA storage. The only downside is that in the sequence-based storage, as I mentioned, you can store 455 exabytes of data in 1 gram of DNA, whereas in structure-based, you can, this is reduced 30%. 30% less data you can store in 1 gram of DNA. So, but still, with this trade-off, we have much better thing like you can read, write, and you can erase the data in structure-based, which is very, very useful thing, right? So probably more and more things. So this has been done in the lab, And Olgika, for example, at UIUC has done a lot of work in this, and there are many other groups. And this is, I think, in future it will evolve, and more people will attract it towards this, and uh, and they can actually contribute in this area. So now I'm going to talk about sequence. There are this is six-step process. First, you encode the data, or your doc file, PDF file, whatever you have, movie, and you convert into the, using data encoding, into the ACGT strings. And these are written using synthesizer machines in the form of synthetic DNA. And then you can store it into the test tube. And whenever you want to retrieve it, I'm not going to talk about a couple of other details inside, like clustering and so on and so forth. But I'm just giving you overall bird eye view. Then you can sort of retrieve it and then do the sequencing and read the data. So there is a software which will actually which has done the encoding part. Now it will do the decoding part, and the software takes care of the errors that may happen, and the common errors which are there, are insertion, deletion, and substitution of bits like ACGT. That is possible, and luckily we have very interesting codes like VT codes and Hilbert codes that have been designed for deletion error correction, right? So there are error correcting algorithms which can take care of all this, and we can actually smoothly store the data in DNA. I'll give you a very brief history of what is possible here in brief history of DNA storage. So, the idea of genetic memory was given by Richard Dawkins in this book, 1986 book, right? Um, and after that, in 1996, the first demo of the concept of DNA data storage was uh, uh, done by Joe Davis, actually, in 1996. So Davis included a 35-bit image of the ancient Germanic rune for female Earth, and after that, so it was fleshing. Essentially, there were a couple of early other attempts, small, storing very small data, like in 1999, 2001. So I would say that a significant work happened in 2012 when Church actually uh, did this experiment. So I'm going to briefly talk about the work of Church and then Goldman. And one of the co-authors at Goldman is Emily, who founded Twist Bios, who is one of the founder of Twist Biosciences 2013. And then Microsoft jumped into this area around 2016, and they also produced one of the product in this area. I mean, the $10,000 prototype of DNA storage. We entered in this area around the same time in 2012, 2013, and we did modified the work of Goldman, actually. And after that, we did some work in 2015 and so on. There are Robert Grass has done a lot of work in 2015. Olgika also entered, and then since then, a lot of work has been done, in this area. And these are some early work. You can see that the red ones are the very beginning work, and these blue ones are which we have done initially. And Microsoft work, which was very very important work, was done in 2016, and then there are many others. Okay, so I'll now talk about the briefly how this works. So in the archival DNA storage model by Church, Gao and Kosuri. So they have this chunk architecture. There are some index bits and some information bits, and the length of the chunk was 115, and number of chunk was 54,899, and they stored 5.27 megabyte MB book, right? And there was no error correction here. So once we saw this, we realized that there is a possibility of error correction. By the time we actually think about a method, there was another work by Goldman, Nick Goldman and his group, and in the Nature. And so here they did some error correction mechanisms, and they also included parity bits, and they used error correction mechanism known as four-fold redundancy method. Okay, so basically this was really interesting work. But and one of the BTech student at our place, Shalin Shah, when he approached me, I said he wanted to do something new. So I said, just implement this paper. And then we realized that the algorithm has certain limitations, that it can only encode easily up to 500 MB data. So 500 MB file. So we improved the algorithms, and using a Gole correcting code. I'll show you with an example what we did actually. And then so this they were able to store data in the order of petabytes. We actually did it in terms of exabyte. Okay, and so we also released a software. This is available, anyone can use it actually. Here, this is the picture of Shalin. Dikshita is my PhD student, earlier PhD student. And so this software is available, DNA Cloud version 1.0, which you can use for storing data into the DNA. Here is an example. We in our improved version we used Gole subcode. We call it. Gole is a very famous. Gole code is a very famous code. We took the ternary Gole code, and this is a roughly chunk architecture. There are certain base pairs for chunk, chunk info, chunk header, and so on and so forth. I mean, this is. Let me show you. So, suppose you have a DICT, ASCII values are this. Then we map these ASCII values to these ternary code words using a table. I'll show you that table later on. And then these tables, these ternary strings, so what kind of data is there, whether it is a text file or video, audio, whatever those things, extensions, file extensions, and some separators. We add. So we actually make chunks of 99 length, of ACGT. So every chunk should be of the length of 99. So multiple of 99. So basically, we have here final string. You see from this ternary. I'll show you how we get ACGT. So suppose these are the strings here. So we have 0. We can assume this. We use this rotation map, which is discovered by Church. So once you look at, you can assume, so when we want to start to encode 0, we can assume any previously nucleotide. Suppose it is A, so 0 should go to C, which is here, right? You can assume anything, but this is C. So next you go to 2. So 2 is here in the table, and now you see the previous written nucleotide is C. So 2 to C will give you, A, right? So A is there. So like that, you can convert these ternary strings. And why do we need this map? This map ensures that no ACGT is repeated. So AA, CC, GG, TT will not come anywhere in the string. And this is important because if that happens, actually, I think there is a question. I will answer the question at the end. Okay. So basically, so these mappings ensure that we don't have homopolymers of, so no repetition of ACGT. Okay. And so this is the final string. These are the DNA chunks of length 99, and we add indexing and so on. So you can see these red ones are index, and the file index and so on. And then these are, synthesized using a machine. So currently, in the current technology, normally people go with 200 nucleotides. Of course, you can go a bit higher, maybe 300, 350, but the standards people are keeping around 200 nucleotides for each strand. So in each piece of information, each chunk actually size is 200 nucleotides. So we implemented this Gole code one into the basically in the second version of the software. And so basically, as I mentioned, so we are converting basically, uh, ternary to ACGT, is 012, 012 to ACGT. Okay. And there was a huge significant improvements in terms of Goldman result and our result. You can see here, in terms of number of size. And one thing you will observe that in their algorithm, Goldman's algorithm, chunk size was fixed, and now we have the dynamic chunk size, right? So which was a really important, because now we were able to do, a lot more. We are able to do less number with less number of chunks and less number of nucleotides. Now we also went a bit further, and we also stored data into the bacteria. In the plasmid DNA, we stored bacteria. In fact, we have come up with our own software, which is, you can store, now you can store your data in your stomach. But we have our software which can actually, you can use this, the first software for bacterial data storage. Okay. So in fact, as I mentioned, we have done the experiment. This was done by, this was done at National NCBS Bangalore with Taslim Arif, and we actually encoded Startup India, Stand Up India, and so last four strands actually were for Startup India, Stand Up India, right? It was 621 base pair. We actually stored data into the bacteria and retrieved it successfully, and then we demonstrated to the Prime Minister. This was the demonstration that we did in 2018. So, I mean, this software, I'll just show you some examples like Indian talents time Israel technology, because we did it for the demonstration of the prime minister. So we actually took their, this text is converted into the ACGT here. Similarly, you can see, I mean, if you we can do the QR code scan, you will see the image of this image to the DNA, this audio and the video. Everything is possible with our software. So I'll show you a couple of recent developments. This was the initial history that has happened, and I talked about some of our initial work. So I think in 2021, Illumina, Microsoft, Twist Bioscience, and Investor Digital, they form an alliance known as DNA Data Storage Alliance. We are also part of this alliance, and now there are right now 37 members. Now this is part of ISNIA. Okay, so here actually right now with this ISNIA, uh, ISNIA, I think some of you might be knowing the Silicon Valley, they do standards for data storage technology. So naturally, this is part of DNA storage is part of the ISNIA, and we are working on so right now there are different board members, biomemory catalog. In fact, I will show you some of the recent developments where biomemory is the first commercial product of DNA storage catalog from MIT, and then a couple of other companies are the board members right now. In fact, I am contributing. I am part of some couple of committees here in Codec working group, and it was a roadmap working group, and DNA data storage Roseton working group. So actually, here we have released the first specification of data archival data storage. I'll show you that thing pretty soon. So another project we did actually was on archival DNA storage with our collaborator Dr. David Smith at Fraunhofer, and this was supported by Indo-German grant to DST DAT. In fact, one of the motivation what we were doing is there is as I mentioned there is a branch in DNA computing known as DNA origami. So one Paul Rotman actually took a M13 virus which infects bacteria. It's around 7,000 base pair long, and it can he folded into the different shapes. So that branch is known as DNA origami. Now, this DNA, there is a birth of DNA origami cryptography also for secure communication recently happened. So motivated by this, actually, of course, we have done some work there, and we have done some work in this direction with David Smith. So, I mean, our objective was we should develop more DNA origami based crypto system, but we have this software 3DNA, which can create 3D structures out of DNA. Anyway, so we are also part of JPEG DNA storage group. So now you see, when we want to look at a specific media, for example, JPG, right? So JPG is a very common image format, right? Billions of images are created every day actually from JPG, right? So whenever we take pictures and so on. So the JPEG committee, which is pretty standard, which is a part of ISO. This is SC29 WG1 committee. So there, Professor Turaj, he is chairman of this committee. So we have, here we are developing the standards. How do we are trying to modify the codec of JPEG and see which one is better for DNA storage. So that the specific format can come for JPEG DNA, and of course there are a couple of security aspects as well. Now I'll show you some recent news headlines actually. So there is a uh, uh, rewritable storage was done first time in 2012. Okay, rewritable DNA storage, and then we have CRISPR-Cas encoding of a digital movie into the genomes of population of living bacteria was done in 2017. This is, you see, image reconstructed from bacteria on right hand side, and this is the original image. All this work is done. And it's a very interesting thing, DNA malware attack in 2017. So basically, scientists converted a virus into, the virus which infects the computer, right? The malware into DNA strands, and those DNA strands were synthesized at the computer, and the virus was activated. The malware was activated actually. So this is the first kind of DNA malware attack on a machine, which was done in 2017. There are a lot of things possible actually. A lot of things are happening. I think I don't have enough time to talk about the security aspects, but there are so many things are happening at the moment in this area. Okay. So our work in 2018 was also appeared in Times of India in 2018 when we demonstrated to the Prime Minister. And by the way, in 2018, Catalog came up with a very nice, synthesizer or DNA writer, and I think they are doing some tremendous work. I am in touch with them, and they're doing some pretty cool work in the DNA storage technology. They're coming up with a single machine for doing the entire thing. This is their 2018 model actually. Microsoft has, come up with. I think I'll take this. So should we call DNA inspired synthetic storage instead of DNA storage, as the real storage seems non-biological? Well, it is synthetic. Yeah, I mean, you can call it synthetic storage. That is also a possible. That is also a term people use actually. So Microsoft in 2019 they have come up with the $10,000 prototype. This is a machine, and they actually encoded "Hello World." These many bits, and it took approximately 1 milligram of DNA, and write and read took 21 hours. With this machine, just a prototype, that they did. In fact, and this is I was talking about Olga's work. They've used rewritable DNA storage demonstration was done. So just like DNA, like punch cards to store data. So this is really, really very, very interesting work that happened in 2020. Okay. Biohackers, the Netflix show was stored in by Twist was stored by Twist Biosciences. In September 20. JPEG DNA also emerged in September 20, as I mentioned, and a lot more work is going on. You can find more information about this at this page. Okay. Uh, Another, this is really interesting thing. Density. So the new twist on DNA. I mean, this, you can actually see the preview just like that. Whenever you look at thumbnail of a image, right, before downloading. So similar kind of technology was developed by was developed in 2021, right? So again, our work on bacterial storage was reported in 2021 in Times of India. Uh, Another very interesting thing happened, as I mentioned. 1 gram of DNA can store 455 exabytes of data. In April 2022, George Church came up with a very natural encoder, which is known as Yin Yang codec system, which where they have achieved actually 432 exabytes per 1 gram of DNA. So really, they have achieved this in the lab. Okay, so as you know, 455 exabytes is the limit, but they were able to achieve this. This is really nice work. Okay. Again, Catalog demonstrated fundamental parallel search capability using DNA chemistry. So this is something another interesting thing. Catalog is doing really very good work in 2022. Uh, Tom team developed a microcapsule of protein that contains one file. This is another direction in 2023. Well, backcam has been invented. So DNA shows images from a living digital bacteria. This paper is really nice, so a biological camera that captures and stored images directly into the DNA. This appeared in Nature in 2023. Biomemory, as I mentioned, this company from France. So they are pretty soon. They are going to release the world's first commercially available DNA storage product. Actually, Biomemory. I think so. You can actually join their wait list if you want to actually buy this, and the cost is something around €1,000. For this product. This is a very recent news for December 2023. And as we were a part of this DNA Data Storage Alliance, and we have released this first specification. Joel from Dell was the chairman of this committee, and so that in the hard disk we have the different sectors. So similarly, architecture was proposed in the form of sector 0, sector 1, and so on. It's really interesting work. It is released now. You can read more about it. This is released very recently, 12th March 2024. There are a lot of companies now which you can look at it. In fact, right now there are 48 companies working on this area. So this is at our lab page. You can find dnaresource.guptalab.org. Some of the work we have compiled here: softwares, research grants, courses, companies, book, and so on. Right. So and about 400 research papers also we have listed there. So I would like to say that now there are couple of challenges still left, and the main bottleneck is still DNA writing, the synthesis technology, which is very, very challenging. As I mentioned, there are three kind of synthesis techniques, and one of them is enzyme based, which is again promoted by Joy Church Group, which showing a lot of potential actually. Okay, developing security is another very, very important thing, and mm, efficient clustering algorithms, optimal codec, and it's still the DNA writing cost is still too much. So I think unless it is at a reasonable thing, people won't use it. So I think now companies efforts from companies like Biomemory, Twist Biosciences, and Catalog. So these some of these companies are trying to actually come up with the actual product with reduced DNA synthesis cost, so that more and more people can use it actually. And of course, you can explore other living organisms such as bacteria, plants, or even humans, right? Who knows? So this is how you store elephants. You see the early CD-ROM contains so many papers, and you see now in a test tube you can store, a lot of information, right? Now I would like to conclude with a comment with Steve Jobs. I think the biggest innovation of this 21st century will be at the intersection of biology and technology, and new era is beginning actually. Okay, so as somebody pointed out for the audience, that we should call it as synthetic data storage. That's normal, and I'm thankful to my collaborators who have done work with me in this area, and of course I support from different organizations: ISO, JPEG DNA, SNIA, DAD, Pulija, DICT, and Microsoft Research. So who have supported some of our work initially, and so I think these are some of my students who have contributed in this area. And I think if you have any questions, you can actually look at a course in DNA storage. We have offered world first course on DNA storage. Around 20 lectures are available online. So the last 2 years we have offered it, so you can get it in this channel. So I think
Yeah.
I'll stop here, and if you have any questions, I think I'm in time, hopefully. So you
Yeah,
can
you
ask.
are perfectly in time, Dr. Gupta.
Yeah.
I have I know there will be a floodgate of questions coming from people. I have
Okay, sure.
one question for to get us started first. So the first question is, I saw you said that 1 gram of DNA can store about 430
55 for 455 exabytes of data. Yeah.
432 exabytes of data, right? In one
Yeah.
gram.
Yeah.
Human
Yeah.
body has how many grams of DNA?
Well, that's really interesting question. I never thought, because we have trillions of cells, right?
Right,
And each cell has around 3 billion base pair, right, of
right.
DNA actually. So yeah, I mean, I think we need to do math. So on the top of my head, I have never calculated in terms of gram, but I think that's an interesting question. I will probably work out this thing for you in the next time. But
Okay, okay, okay.
yeah,
No, I can
yeah,
study.
this is really
So
interesting question. Yeah,
yeah,
so, yeah,
we can say that as a human, we are capable of how much storage actually.
yeah, yeah. That's really
And then
interesting.
the follow-up question would be how often the DNA's update or change. I don't know that actually. But I'm curious, basically,
You
in
mean
a,
to say inside our body?
inside the body, right?
Yeah, actually, so it evolves. There are mutations, and some mutations happens. You know, some mutations are known as good mutations. Some mutations are known as bad mutations,
Right,
and DNA evolution happens over a lot of I mean, actual evolution for good takes a lot of time. But actually, you see, on the other hand, suppose you get some infection. Okay,
right,
say infection of say HIV or any other infection, say the for example retroviruses such as this that common cold virus, right?
right.
So we know that now 2% of our genome is made of these viruses actually, right? So
Mhm.
that anyway, when they want to activate, they actually integrate our inside our cell, and then that their DNA is copied, right? So of course there is a change in our DNA, but how long it happens, and normally bigger change takes a lot of time actually. On the other hand, now if you go to the epigenetics level, so suppose we smoke, right?
Right.
So the methylation frequency will increase inside our
Mm.
DNA, right? So it will hypermethylate, and that passes to the next generation as well, right? So somebody who, maybe third or fourth generation who has never done any smoking will get a lung cancer, right? That is a possibility.
Yeah,
So,
because of the,
so it's a really complex thing, and yeah, many things are there. And I
yeah,
would say that it's still even so much work has been done in the human genome projects, and still I would say that though we understand bit better, much better as compared to year 2000, 20 years now, but still a lot of things are still we are trying to understand. Yeah.
yeah. And my last question is, since this is Silicon Valley Quantum Computing Group, right?
Hmm.
How do we, is there any work done on um, encoding in terms of qubits or in using qubits in this realm of things instead of regular bits?
Yeah, so actually there is one very famous paper work done by Roser Penrose and Stuart Hambrough. I
Okay.
I hope you must be knowing Roser Penrose was yeah, he's a very famous mathematician, and Penrose tiling is very famous tiling on his name. He got a Nobel Prize also a couple of years back, and I met them actually in 2003 at Quantum Mind Conference. So their proposed model is that we have consciousness because of quantum computer inside our brain. Of course, people are debating about it, saying that inside our neuron there is a tubulin protein which can act as a qubit.
Okay.
It can be in two states, right? So, and they propose a lot of. I think Stuart Hameroff has written a book also on this, which is available.
Oh,
So a lot of interesting ideas, and of course, it's a debatable thing, but yeah, interesting ideas, I would say. And also, I think some people have observed that these migrating birds use some sort of quantum algorithms to navigate. I mean, of course, so all these
yeah.
things, one can, yeah,
No, I
yeah,
think the entire area, and correct me if I'm wrong, of natural computing, which basically should include DNA as well as quantum and the same.
quantum, yeah, is,
Yeah,
yeah,
yeah, is
yeah.
evolving. I'm gonna open the floodgates. So, who wants to? I see Prakash has a question. Prakash, do you want to come on the camera and ask the question, or you want me to read it?
Yeah, I can come on
Thank you.
camera. Yeah. So my question was obviously I think you already have uh, indicated about the natural computing. So
Hmm.
my question was about the uh, how do we compare because it's at the end of it it is all what you call synthetic. So the
Hmm.
question is of encoding decoding and
Yeah.
you use ACTG the DNA storage.
Hmm.
So
Hmm.
in quantum, electron itself is the storage, photon
Hmm.
itself is the storage.
Hmm.
So how do you compare storage technologies in terms of their capability of
I,
storing higher and higher uh,
yeah, so
exabytes
I
or
think, yeah,
petabytes or whatever?
yeah, that's interesting thing. But I think, to the best of my knowledge, uh, there are some work in quantum storage, right? But I think I've not seen a comparison between these two yet. Maybe somebody has to do this actually, in terms of the capability. But certainly, I mean,
Mhm.
that's a really fundamental question. I think there must be a connection, because you see, ultimately,
Mhm.
we are also, because in our body, for example, DNA is one
Mhm.
way we are storing data, but then there could be other possibilities as well, right? This is just we are looking at one window, so there might be
Correct.
a possibility, and I think that is something we need to explore further. Actually, to the best of my knowledge, I have not seen a
And
work
does
on comparison work actually between both. Yeah, but yeah, that's something
is there anything in the is there anything in SNIA you came across where there is other than the DNA storage that you have observed which
No,
is
right. So
uh,
no, in
similar or
ISNIA,
better?
they are mainly working on this technology that we are using currently, right? I mean, all these hard drives and everything. So that's the main thing. DNA
Okay.
storage they have taken up, I think, in 2020, from 2020, and now it is. I mean, so we are doing a lot of work in DNA storage right now to make it, And I think if you go to the ISNIA website, you can find a
Mhm.
lot of interesting articles and videos, some more information about it.
Sure, I'll do that. Thanks a lot and appreciate your time and a great show.
Thanks.
Dhawal, I see you unmuted. You have a question, sir?
Yeah, I do have a question. I wouldn't like to come on camera. not yet prepared for that. So sorry for that. It was excellent presentation, Dr. Gupta
Thank you.
and Gupta ji. So I appreciate that. A couple of questions, and I'm coming in this from new, so please treat my questions as a newbie.
Mhm.
How stable is this DNA storage in terms of let's say heat or cold variations? Do we have to keep it in? Currently, it is in lab environment, so we understand that. But the question comes is that when it gets commercialized sooner than later, like within 5 years or so, how stable
Mhm.
it is this?
Yeah, so this is a great question. So I would say that actually now scientists are developing different kind of DNA containers. Okay, and to make sure that it is as safe as your hard disk kind of things, right? But because even if so, for example, if you put your hard disk in the fire, of course it will go off, right? I mean, all
Mhm.
the data will go away. In the similar way, right? I mean, all the DNA is also I mean, if there are some, there are environmental conditions may cause some damage. Now, on the other hand, just like in the hard disk, there are algorithms which, up to certain extent, protect you, right? I mean, if there is error or something, it will correct the errors, and you are able to still read the data. Similar things people are developing for the DNA stories, right? So, I think up to certain threshold, everything should be fine, but yes, beyond extreme weather or extreme things, but on the other hand, you can observe that people have found DNA 10,000 years old, right? And they are able to
Yes.
extract that. Yeah, so obviously, I think it is probably more natural. How much information you can retrieve, that depends on how much replication. I mean, how much replication of DNA. Because you see, one thing people do, they do a lot of PCR. So do a lot of PCR and
Mhm.
create a lot of strand. And so I hope you have sufficient DNA to reconstruct the whole
Yeah.
data.
Yeah,
Yeah,
one can have a redundancy as well because and
yeah,
then we know that tardigrades survive space as
yeah,
well.
yeah,
Tardigrades. So there is that's why the reason is that you can have a very compact memory bank and
yes,
you don't and very resilient as well without
yeah.
any extra care. But thanks for
Right.
answering that.
Thanks. I think there's another question.
Yeah. Anybody else has a question? Okay, Abhishek, go ahead. You want to come on the camera and ask the question, sir?
Uh, yeah, no, thanks, Tapash. Sorry, I just came out of bed and I joined this early in the morning. Dr. Gupta, so thank you so much. Actually, this is a very interesting topic. I wasn't even aware of this entire space being there in the post. Actually, messaged that there is an interesting presentation on this topic. It was actually very eye-opening for me. So thank you for sharing all of this details. Just
Thank
a
you.
curious question. Um,
Hmm.
I, I was trying to follow the presentation. It was a little fast for me to be able to catch everything. But the one thing that
Hmm.
I was interested is to understand if there is a, you spoke about the failure rates, like hard drives, SSDs today, they have a failure rate at certain, and they have a life
Correct.
on
Correct.
how long
Correct.
they last.
Hmm.
And also in terms of read write, there is always a challenge, like how quickly can we read and write data.
Correct.
With this approach, yeah, how comparable is this technology or this approach compared to what we have today?
Yeah. So I think I will say there are two parts. First is that right now technology people are focusing on archival storage. So basically only and as I've shown you in terms of it all depends on the speed of the this DNA writer and DNA sequencer, right? Those machines actually, right? Now there you can do certain parallelism thing, but obviously the technology of storage that is available to us today in the modern technology, it is not up to that level yet. But now we want to say how far. I mean, so people are trying to speed up. And as I mentioned, for archival storage, it is fine. I mean, you store it, so maybe it takes 5 days, 10 days, or whatever to store, write it, and retrieve it. Sequencing is fast, but writing is a bit lengthy. But what I'm saying that actually, rewritable storage, which is engine based, actually is better actually. So that power portion of technology is still in the lab, and not nobody has done any commercialization on that part. So I think that is still open. I think as I mentioned, it was started very recently, 2022 or something. So a lot more work is needed in that direction. People are trying. There are different groups who are trying. I know a couple of groups in Europe, as also in US, and they are trying to develop that. So I think in the standard committees, we are trying to define what could be the reasonable way of doing these things. What should be the reading speed or writing speed and so on and so forth. So we are trying to develop those things. Obviously, it is not comparable as compared to the current one, but it will take some time to really
It,
do more work in this area.
it's dependent upon the algorithm that's being used to encode.
Yes. Yeah. So
Okay.
not only the algorithm, actually the machine, because you see, synthesizing DNA is a challenge. What happened because of Human Genome Project? People were reading DNA, so that technology has matured well enough that we have very good machines. But on the other hand, in terms of writing, there are three approaches. Right now, the most popular one is chemical-based synthesizing, and I think that is, not that good as compared to. Of course, it does the work, but enzyme-based is much better. And I think a lot more work is needed. So I think, as I mentioned, catalog is coming up with a very nice product in this area, and I hope so. You soon some of you will see that commercial product available sometime, from there.
Thank you. Thank you so much.
Yeah, so I saw somewhere, Dr. Gupta, that it took 21 hours to write to the DNA, right?
Yeah,
Is that
that
the
was yeah that was 20 by Microsoft experiment. So I
experiment?
think
Yeah.
I can show you. Yeah, that slide is here. Yeah, so basically what happens, this is a single prototype, right? So single machine doing everything together is again a challenge. One way is, you there are synthesizers available, there are sequencers available, and you use a separate things. Then there is a software available at somewhere, right? You do independent things, but otherwise, having one box, having everything, inside, uh, different kind of things are needed actually.
No, I totally understand. And I did some work with very basic work with DNA synthesizers in early 2000s. So I have
Yeah.
some basic idea, but not as much. But I can relate to it, how much complexity and how much of other liquids and other things are needed for doing
Right,
something
right.
like
And
this.
I think it requires standards, and that's what we are trying to do. So making standards in everywhere, right? So that we know what are the efficient way of doing
Yeah,
things,
yeah,
Yeah.
yeah.
Can the archivals adopt the warm write once, read many times, warm drive standards? Adopt for this DNA
Uh,
storage?
yeah, you can read it. Basically, what happens, because you can do the replication easily, right? So I mean, that is a possibility. Yeah. So because see what happens, you can have multiple copies of DNA because replication is easy and fast, right? Of the DNA. Same thing even with bacteria. But of course, the size of the amount of information you can store in bacteria is limited by the plasmid size. But otherwise, what I'm saying, that replication is easy, right? PCR we do so often, right? So once the
I
DNA
see.
is there, we do PCR, right? So that is, so
Okay.
then, I mean, you take some portion and do it. So normally what happens, actually, I gave you very simplistic view. So once we get the single strand DNA, then we do the replication, we do the PCR and a couple of things. And while reconstruction, we do clustering algorithm. So people are developing efficient clustering algorithms, which can actually, so basically what I'm saying, mathematically, you can think of there's a single DNA strand, and then out of that, there are, say, maybe 300 DNA strands out of single DNA, all copies of the same strand, right? And then you store data. So when errors will happen, so now you do the clustering out of those strands, and do after that doing the clustering, you do remove the errors and get a single strand and then decode it back. So there's a lot
I
more
see.
processes involved in that. So I gave you a very simplistic view right now.
No, no, no. I get it. Thanks a lot. Appreciate it.
Mm.
We are at 10 a.m. So I know there is a lot. Maybe we'll invite Dr. Gupta once again later on. But really, really would like to thank you, Dr. Gupta, for taking your time and joining the Silicon Valley Quantum Computing Group. Maybe I should change it to Silicon Valley Natural Computing Group because it's more apt now.
Yeah.
Really appreciate it. Thank you, everyone.
Thanks. Thanks. Thanks, Tapash. Thanks, Tapash.
Thank you everyone. Bye.
Suppose that is called scope creep.
Scope creep. Thank you, Prakash.
Thanks,
Yeah,
Ankush.
thank you.
Great.
Thank
Thanks.
Thank you so much.
Thanks, everyone.
Thank you.